So now that we've covered the principles of mutation calling, we can take the next step of trying to annotate what the effect of a mutation might be. So as you remember from the last module, mutation calling just generates the genotype of the tumor, or what are the DNA changes that are mutant bases that are going to be in the tumor as opposed to a normal sample. And so often this is stored in what's called a variant call format, file format, or VCF file, and it just contains information about the chromosome, the position, what the original base was, and what the mutation is, along with various other quality control metrics. However, this doesn't tell us what the mutation actually does, it just says there's a mutation there. So the next step is to annotate what the mutation might be doing, and then visually interpret the mutation to see if there's any convincing evidence for it having an effect. So one tool to annotate mutations is called Cravat, and it has an online website where you can actually submit mutations in VCF file format. There are other annotators for mutations like Variant Effect Predictor or VEP, but we're going to be focusing in on Cravat as it provides as an easy interface. So you can see that the typical information like you had from the VCF file about the chromosome, what the actual DNA change is. You can see other associated cross-reference information from various databases about whether there's any disease associations. And then you can sort, filter, and look at your variants to better understand what mutations you've actually seen in your study. And then lastly, we'll be more looking into Cravat, into the homework as a way to get a better feel of what mutations look like from a cancer sequencing study. And so you'll see more of that to come. So another way of understanding mutations is to look at them on protein structure. So often protein structure relates to the function of a protein or what it does. This is because protein structure gives you information about the shape of a protein in three-dimensional space. So you can see this by the ribbon diagram of the protein run X1. And you can think of proteins as molecular machines that often rely on shape to perform its function. So you can see here that this protein is actually binding DNA because its shape perfectly matches the DNA which it's supposed to bind to. And then so one might imagine if this protein structure or the shape of a protein provides information about what it does, we could potentially use the information about the location of a mutation to interpret the effect of the mutation on protein structure. So this is a case for, you can see for 3 mutations in the gene run X1, where I've highlighted where the mutations occur as these little green spherical representations of amino acid residues. And you can see actually there's a common theme here. All of these mutations here, here, and here actually occur at the DNA binding interface of this transcription factor. So this might immediately lead you to think, well, if they all occur at the DNA binding interface, maybe they have an effect either on the strength of DNA binding or how specific its DNA binding is to a specific sequence of DNA. And this kind of general idea of using protein structure and visualization to interpret mutations has been more formalized by use of clustering algorithms that cluster the position of mutations in three-dimensional space and protein structures to identify areas of the protein that have very high clustering patterns of mutations, indicating they may be doing a similar function on the protein. So another type of approach to interpret mutations uses what's called machine learning. So in this setup, you have two different types of mutations. You have one that is your set of mutations that are putatively bad or cancer promoting, that are driving the cancer. And then you have another set of mutations that are fairly neutral or aren't providing an advantage to cancer cells. And the goal of machine learning then is to encode these mutations or variants with a set of features that describe their properties. And so in this case, with the method CHASM, it actually uses 85 different features that describe the properties of a mutation. And this includes what is the sequence like, what is the evolutionary conservation across species, and so often this is a good proxy for whether a mutation may have a functional effect on a protein. The predicted protein structure, which we had just gone over, can be important for interpretation, and additional information about the annotation of key regions of the protein that are important for its function, along with many other different features, including physiochemical properties of a mutation. And so once you have this set of features, you can train a model to discriminate between these different types of mutations. And so in CHASM, this was done by training a machine learning algorithm called a random forest to distinguish between these two types. Once you have trained a model, you can then ideally predict new mutations which you've never seen before, to understand whether they're more likely to have an effect and be sort of cancer promoting or driving cancer growth, or being neutral, having more of a neutral effect. And so what happens is, if you have a new mutation, you can still calculate all of these 85 different features that you had before, but you just don't know whether it's a cancer promoting or neutral mutation. Then with that, machine learning from its learned trained model will produce a numerical score reflecting the likelihood of being a cancer, in this case a cancer promoting mutation. And for the purposes of a random forest, the score is actually composed of many little individual decision trees, where each decision tree actually votes on whether it's a cancer promoting or neutral mutation. And so you just sum up the different decision trees to come up with a score that, in this case, perhaps is 5 out of 6 in this toy example, supporting that's a cancer promoting mutation. And if you score many of the mutations in a large study, say you may have thousands or even millions, you can't look through each one individually. You can try to prioritize and rank by a score that would indicate that they're more likely to be relevant to begin with.